All right, well, I am happy to be here with you today, sharing a very quick snapshot of some of the research that we've been doing over, I would say, the last 7 or 8 years now, mainly in the Olympic National Park. And I want to thank all of my collaborators who have helped make this happen. I always like to credit people before we start the presentation because it really does take a team to make the dream work. And I would not be able to be here without any of these people. And Dr. Jen Kane, who is part of this presentation, but for time wise will not be joining into the presentation with me, has been wonderful in expanding a lot of this research in the recent year. So I am going to talk about some of our canopy soil research. So we can go to the next slide. And so the sites that we're going to be focusing on today span across two different areas in the Olympic National Park. They are both within the lowland riparian areas, and one of them is right off of the Hoh River, and one of them is right off of the Queets River. These are both two old growth stands with similar species composition, both dominated by old growth bigleaf maple and old growth Sitka spruce. Western hemlock are sprinkled in there as well. Understory is largely swordfern dominated with some other things in there. If you're familiar with the Olympic National Park, it's what a typical old growth stand would look like in the lowland areas. So we can go to the next slide. So one of the things that really got me excited on this topic of research when I really started exploring and understanding what canopy soils actually were, which we'll go into in a second, was that when we think about what forests provide us and how forests function, they have a lot of different means to not only provide for society, but they benefit our planet to a immense scale. And so when I walked into these old growth forests and realized that canopy soils were a thing, it kind of became apparent that they're not really considered in forest function in general. And for the last 40 years that they've been researched, they've widely just kind of in general been ignored by the scientific community. And I don't know if that's necessarily because people choose to ignore them. It's just because the science is in its infancy. And we really need people to advocate for the importance of these ecosystems, especially when we think about forest management and the ecosystem services that they provide. So we can go to the next slide. So, what are canopy soils? And I apologize for this because I did not think about clicking through bullets on a presentation that I was not controlling. So, if you want to just give a couple clicks, it will lay out what it's about to say. But canopy soils, yeah, that's good. So canopy soils are thick mats of organic matter that form on tree branches. From this picture, hopefully you can see the scale of this. This is not just like a small mat of organic matter on tree branches. Canopy soils, they form from the bryophytic community and other higher epiphytes. So the mosses, the licorice fern. I've even seen like a 5-foot spruce tree growing up in a maple canopy soil mat. So there's a lot of different species that establish up there, but most dominantly the bryophytes and the licorice fern. Over decades, they form this mat on the branches, which then not only accumulates over time due to the life cycle of the bryophytes and other higher epiphytes breaking down and decomposing, but what also happens is there's a lot of litter interception. There's also a lot of other animals, for example, that go up there and drop things off, and just over all of these years of litter fall and moss establishment, the decomposition over time forms into these thick mats. And I just want to give this kind of a perspective that I have climbed, specifically, this is one of the trees that I've climbed. They have canopy soil mats that are over a foot deep. So that is really rich organic matter on these tree branches. And we can go to the next slide. And so, one of the things that I first saw when I started climbing up into these magnificent ecosystems, I honestly feel so blessed that I get to participate in this research, is that when you started kind of sampling and parting through these canopy soil mats, which you can see here in both the pictures, I saw these roots and I was just like, wait a minute. What are these roots from? I was just a young aspiring scientist at the time. I'd never really been up in a canopy soil environment. So when I started asking my mentor, what are these roots? Like there doesn't seem to be any plants that are large enough to have roots of this size up in the canopy. And then she said, oh, those are roots from the host tree. So I was like, wait a minute, these are specifically big leaf maples that we're climbing. So I was like, okay, so like in the ground, the trees are actually growing adventitious roots out of the branches into this canopy soil. Like my mind was already blown at that point in time. And so to take it even further, I was like, okay, there's roots up here. Like, what are they doing? What fungal organisms are they associating with? What does this mean for these forests in the face of changing climate? And the answer to that question was, well, we really don't know because nobody's ever looked at that. So some of the questions that we've started formulating and answered over this long project. Like I said, I've been working in this ecosystem for about 8 or 9 years now. I shouldn't say long project, especially if we have some people who work on LTER sites here. So I would say that I established this project, and for me, it's been a long time. But for other research projects, relatively, it's not that long of a research project. But some of the questions we started asking was, okay, well, if there's roots up here, do they have mutualistic reassociated fungal communities? For example, mycorrhizae that can uptake nutrients and provide nutrients for the plants in exchange for sugar. That could mean that these host trees were really benefiting from their canopy soils. But in order to do that, they would also have to have a flux of available nitrogen and phosphorus for the plant to uptake. So what about that? And then it just started kind of expanding from, okay, well, canopy soils may be providing a benefit for the host tree, but also what do they provide at a forest stand level? So when we step back and look at canopy soils in these ecosystems, they are hypothesis-wise benefiting these host trees, but they are also likely contributing a huge extent to these ecosystem processes. And we've really only started to brush the surface on understanding how they do that. So throughout that, we've also started collaborations on looking at other biodiversity measures and overall just collectively trying to understand how important canopy soils really are for these ecosystems. And that steps outside of canopy soils as, for example, a habitat for endangered species like the marble murrelet or threatened species like the spotted owl. We were kind of looking at the unseen, like what can you not see that really drives how these ecosystems function. So we can go to the next slide. So let's go back one more. We skipped one. Yeah, thanks. So now what we're going to do is we're just going to kind of go over a crash course of all of these different projects and the findings that we've had because they're really exciting. And within the time that we have, we don't really have a lot of opportunity to dig really deep into the results, but I'm happy to answer questions. You can email me after this if you have anything that wasn't answered. But so the first thing that we did in these trees was we wanted to look at the fungal establishment, so the mycorrhizal establishment in these roots. So again, just to repeat, mycorrhizal fungi attached to plant roots, they expand out, they uptake nutrients for plants in exchange for plant photosynthase, so for sugars. And when you assess the structures and the amount of structures within roots, that can give you some inference to how much energy is being put into these interactions. And also it will just tell you, for example, do we have more fungi in the forest floor level than the canopy level, which starts to answer some questions and allows you to expand more questions. And so when we started looking at these fungal structures, which you can see in some of the images, so if people are not familiar with staring at tiny little roots under a microscope, what we see here is we have roots, the structures that are fluoresced in green or stained in blue or kind of have a darker pigment within the root. Those are mycorrhizal structures. So overall, what we found was I hypothesized that there would not be as much fungal establishment in canopy roots, for example, due to dispersal limitations. But this is one of those cases where having a hypothesis be proved wrong was actually really excited. We found that there was no significant difference between the fungal establishment in canopy roots and forest floor roots, which was just saying that these roots are associating with just as much mutualistic fungi in the canopy as they are on the forest floor. There was a slight bit more on the forest floor, but it wasn't significantly different. So to take this even further, we can go to the next slide. We started looking at these root-associated fungal communities and if they were different between the canopy environment and the forest floor environment. So this isn't only looking at beneficial fungus, but this is looking at other root-associated fungi that may be beneficial at certain times of the year, may be dormant at other times of the year, really just trying to get a collective understanding of these fungal associates in their communities and how they shift. And so this is a PCOA plot, and this is just an ordination in space supported by significant statistics, which shows that we have a clear line between the purple, which are the canopy soil communities, and the green, which are the forest floor communities of these big leaf maple trees. And even further, when we dive a little deeper into the canopy environment, we can see that most of the communities that were from the host site were significantly different than the canopy communities from the Quees site, which starts to build upon this story that we are exploring about dispersal limitation. And we can go to the next plot or the next slide. Yeah. And then from this, we stepped out of comparing the floor and the canopy to just looking at the canopy and what would happen to these fungal communities if we started seeing wetter and drier conditions, because fungal communities can actually shift over different times. For example, in times of drought, you may find fungal communities that help plants acclimate to drought stress. And we really wanted to understand, okay, well, are these canopy environments potentially acting kind of as a resilience compartment for these host trees, or what happens if these canopy soils are pushed past their threshold? Is the host tree going to suffer? And so by implementing these experiments at the canopy level, we had conditions that diverted half the rainfall and conditions that doubled the amount of rainfall to compare to ambient conditions. And if we go to the next slide, what we can see is, although within the PCOA space, it wasn't as clear as a distinguishment between the differences, the statistical tests did support that in canopy soil environments, the root-associated fungal community composition was not only dependent on the rainfall treatments, but it was also dependent on spatial location and the seasonal sampling period. So that just supported what we found at ambient conditions, but with the canopy level only, that these fungal communities and canopy soils are impacted by wetter and drier conditions. But because we don't research canopy soil dynamics as much, we don't really know what that means for these forests and for managing them. Next. So and you can just click through this really quick so we can get the list out. Yep, that's good. And so within looking at the species diversity of these canopy soils, we had a lot of species that were of interest that may have really important functional attributes that we just don't know about because they've never been discovered. So these species, for example, like Mycena, that's usually a decomposer, but there's been research that's shown that it can actually benefit plants at the root interface. So what's it doing in the canopy soil systems? We really don't know, and we should find out because it could be helping or providing a detriment to these canopy roots. And the collection of the other species that we see on here, for example, the dark septate endophytes, those are often species that will help plants, for example, when there is drought stress. So we were seeing a lot of those in the drier months in the canopy roots. We can go to the next slide. And so we can click through here. To just provide a quick conclusion on this, we have obligate mutualists in both canopy and forest floor soil environments. The fungal communities associating with these roots are significantly different, and the rainfall treatments impacted these fungal communities. Cool, we can go to the next slide. And so we'll just go really quickly through these last couple slides. And again, anybody can follow up with me with additional questions. So we then went to compare the rates of nitrogen mineralization, phosphorus mineralization, same experimental design. So at first we looked at ambient conditions. So we can go to the next slide. And we actually found that in different seasons, especially in these latter two seasons, there was a lot of anomalies when we were comparing the amount of mineralized nitrogen and also the amount of mineralized phosphorus. One of the things that stayed pretty consistent throughout different times of the study was that if there was an immobilization or very little phosphorus or nitrogen available at the forest floor level, there was always available nitrogen and phosphorus at the canopy level. We can go to the next one. And so this is the treatment level. It stays pretty consistent with the different findings. I'm just going to skip past this section so we can get to some of the new findings on the horizon. So skip, 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 skip. Okay, so this is another important finding that we found. So we found that overall canopy soils, they contribute to the forest level nitrogen and phosphorus mineralized pools. We also did a greenhouse gas study where we found out that this is just for maple canopy soils, that they contributed 4% to stand level carbon dioxide emissions, and they actually contribute 4% to the methane sink. But the thickness of the canopy soil mats actually impact those methane fluxes. So this is something that we're preparing right now and analyzing a little further, but this isn't considering, for example, Sitka spruce or any of the other canopy soils that exist in this system. So we're actually going out there this weekend to kind of try to get a proxy for that, to kind of understand what we may be missing if we don't consider these. Next slide. And so overall, there's a diverse, functionally significant microscopic community in the community in canopy soils. If we go to the next slide, this is some hot off the press research. For example, so we looked at that dispersal between the two sites and between canopy and forest floor levels, and we found those differences. So what this study did was actually zoomed in on a smaller sample size, but went to different branches. And so, for example, we found that soil respiration overall was higher, which you might expect organic matter in the canopy, was higher in the canopy than the ground. If we go to the next slide, we also found that there was really rare nematodes in the canopy, and the diversity decreases or increases depending on branch height. Next slide. I'm almost done, I promise. And in the soil, so I looked at root-associated fungal diversity with the roots. We also looked at this in the bulk canopy soil and found that bacterial and fungal diversity was highest on the ground and decreased with branch height. Next slide. Next slide. So overall, we can just give it a final click. Canopy soils should not be overlooked in these ecosystems when you're thinking of forest management because adventitious roots associate with mutualistic fungi. They are a nutrient reserve for the host tree and epiphytes. They contribute to forest level nutrient dynamics. And overall, we theorize that they may be a resilience compartment for these old growth trees, especially in a changing climate. And if you're modeling ecosystem processes, whether it's for management or climate change scenarios, we have been emphasizing that if you're ignoring this canopy soil compartment, you're ignoring what we've been seeing to be about 3 to 10% of different pools within this ecosystem, and that's just for maples. So that being said, we can go to the final slide, which is the question slide. I don't know if we have time. Oh, wide diversity of mushrooms that we were finding up there and identifying as well in the sense of biodiversity measures. So yeah, overall, I would be happy to answer questions if there's time. Send me an email if there's not. And it's always hard to get through so much exciting science within the 12 minutes, which I'm sure that all the other presenters also feel the pressure of. So thank you for keeping up with me. I appreciate it.
All right. Yeah, we're a little over, but that's okay. We do have one more talk left, and there's like 10 minutes after that. So we're okay. Couple questions. Robert from DFW, does soil canopy development help define old growth trees more precisely?
Yeah, so I would say that it does help define old growth stands. Maples have an affinity for moss establishment, so I wouldn't say that as soon as you see a mossy mat growing on a maple in a rainforest, that it would be categorized as old growth. But when you're driving through the peninsula and you see those large trees that have the draping moss and the thick mats, whether or not they're in an old growth stand, they're going to be an old growth maple. So yeah, we try to do a study where we actually looked at the establishment and how long it took those mats to reach the depth that they get, but we have so far not gotten funding for that. So hopefully one day we'll be able to answer that more stringently.
Okay, one other question I see in the chat. It's kind of a long one, so you might look in the chat and see. Do you think the difference in fungal communities in the Hoh and Queets trees could be explained by differences in microclimate for these two areas? And then the second question, do you have a different explanation for the difference in fungal composition in these two locations?
Yeah, so I think that the difference between the fungal communities and the Hoh and Queets trees, it could be explained by microclimate differences. We're teasing apart some of our microclimate data between the Hoh and the Queets site, for example, like soil temperature and soil moisture at the canopy soil level weren't really significantly different in their shifts. So we actually hypothesized that it might be based more on, for example, what species might be dwelling up there at any point in time. For example, if certain species enter these canopy soil branches and, you know, make a habitat there, they might be eating different types of fungi or digging through different types of soils and then dispersing the spores up there. There's just a lot of different factors that can come in on what allows fungal communities to establish. For example, a bird could eat something that has a mycorrhizal spore on it one way and it happens to land its poop on that canopy branch of that tree, and then that species is there, but it might not be in another tree. So a lot of questions to be answered there, but I would say that it's probably driven more by organismal interactions than it is soil microclimate. And then do we have explanations for the different fungal composition in these two locations? We are trying to do more diversity studies and also sequence, for example, the organisms that might be associating with the bryophytes, because they're non-vascular plants. So we're also interested in seeing, if we can see a pattern of what's associating with plant foliage, because maybe it's dormant, and then when the plant dies or lands on the branch, it may distribute some of those species there. But overall, I have the best answer to your question. We still don't fully know, but we do know that they're significantly different in certain areas and that they're somewhat linked to different nutrient concentrations, and that mostly our hypotheses are always linked to these dispersal limitations, especially with that recent study where we saw those decreases on the branches. But I will note that even though we saw a decrease in diversity with branch height, the functional impact of canopy soils and like the functionality, it was always higher in the canopy, even if there was less diversity, which kind of reflects on that abundance. What's the word I'm looking for? Redundancy. So, you can have a really high diversity, but there's some functional redundancy. The canopy seems to have a lower diversity, but higher functionality. So.
All right. Let's move on to the final talk. Thank you, Karina. That was a very great presentation. If you have any other questions, feel free to leave them in the comments, and Karina, you can address them directly there.